2-day international webinar on COVID-19 impacts, preventive measures, and best practices. On the morning session of the second day, we have with us Dr. Ria Bosch. She has connected with us from University of Texas, Dallas. So, dear participants, as I mentioned earlier that in the previous session of yesterday, we have 3 lectures followed by Dr. Nirmallo Dasgupta, Professor Dhrubajyoti Chattopadhyay, and in the afternoon session, there was a session by Dr. Sumita Goswami. As we have promised in this session, the morning session, Dr. Bosch has been connected with us. I have a short introduction of her. She is now the postdoctoral research associate at Department of Physics, University of Texas at Dallas. She had done her graduation and master degree from University of Calcutta, done his PhD in inorganic chemistry, material science particular from Indian Association for the Cultivation of Science. She has several awards and accolades in her name. And among them, the very prestigious Alexander von Humboldt Postdoctoral Fellowship, Germany, CEA Enhanced Euro Talents, Mary Skolandri Puri co-found postdoctoral fellowship, SABIC postdoctoral fellowship. She has associated with several positions before she joined at University of Texas. In numerous publications in her name with high impact factor journals, above 25 publications, and overall impact factor of above 200. So she has the overall citations around 800 and many peer-reviewed journals. And the research interests of her are the fundamental understanding of charge carrier dynamics in a variety of semiconductor materials, application of semiconductor nanocrystals and thin films. So with this brief introduction, I would like to request our Vice Principal, Dr. Sanjit Kumar Das, to welcome her on our college's behalf. Then Dr. Bosch will carry on. Over to you, Dr. Das.
Thanks, Dr. Bhadra. Good morning and welcome to this second day of international webinar organized by the Department of Physics and IQAC of Bhairapangi College. Hope you all have enjoyed the first day of this webinar. And now in this first session, it's my pleasure to welcome and invite Dr. Ria Bosch, Postdoctoral Research Associate, University of Texas at Dallas, for her presentation. Please, Dr. Bosch.
Thank you. Good morning, all, and thank you very much to the organizing committee for inviting me to present this talk. I hope you can see my screen. So I will just continue with the presentation. But before I do that, I would like to say a few things that COVID-19 is not actually my research subject. So what I will give you is more of a layman's idea of COVID-19. And I'm sure that many of you have seen the information that I will show you here before. But what I have just tried is I have tried to gather as much information as I can to give an overall better view of what is happening around here. And before I go to COVID-19, the subject itself, I would again like to say a few words about, sorry, my screen somehow doesn't work, so I will try to share it again. Yeah, it works now. So I would like to give a brief introduction about what I work on, which Debabrata already mentioned, that I work on colloidal semiconductor nanocrystals, and my work can be actually broadly divided into 5 parts. So first is I try to control the crystal growth in solution, and I synthesize them basically in solution phase method using a typical setup like that, which involves a 3-neck round bottom flask. And we do most of the reactions in inert atmosphere, and we use to monitor temperature, because most of the reactions are in takes place in high temperature. So use a temperature controller to monitor the temperature we are doing the reaction. So the first question is why I'm working on semiconductor nanocrystals, and why is at all anybody working on them. So we all know that semiconductors have a certain band gap, and when we are actually talking about semiconductors in nanoscale, then even for one particular material, if you just keep on changing the particle size, it will actually change the band gap, which will be inversely proportional to the size of the material. So for these materials, the valence band are full of electrons. So if you shine light or give any kind of energy to this valence band electrons, it may jump to conduction band, and it stays there for a certain amount of time, which is known as the lifetime of, which is characteristic of one particular material. And after that lifetime is over, that electron may come back to the ground state, and it recombines with the whole, and it releases the excess energy. So if this excess energy is released in terms of light, then we actually can get emission from these particles. So now if we I do not know why I cannot change the, okay, here it is. So basically, where if we just keep changing the particle size, we can change the absorption and emission, and thus we can get the emission tunable in the entire visible window. And not only that, we can also get emission in NIR or in UV region. So that's what I am basically interested in, semiconductor nanoparticles that emit light. So further, what we can do is that I also do shape control, and I try to form heterostructures between two different semiconductors or a semiconductor and a metal, as you can see in this picture. So when we form a heterostructure, basically we are connecting two different materials together. So in one particular structure, you can get properties from two different materials. Next, what we can do is that we can change the composition of the material to tune their light emission. So basically, as you can see in this picture, we can have blue emitting zinc selenide, and then we can either change their composition. And here we mostly change the composition of the material in a way so that it does not change their band gap, but it inserts a certain amount of foreign material into the band gap in a way that it creates a state in between the valence band and conduction band. And so it can modify the emission from the host nanocrystals. And you can see if we dope zinc selenide with copper, we can get green emission. If we dope with manganese, we can get yellow emission, and so on. And then we have also monitored the presence of surface and structural defect states. So as I have said that these materials have valence band and conduction band, and usually the emission we get is in between these two channels. But sometimes it happens because of structural defects, there are states that gets incorporated in between valence band and conduction band, which can actually capture the electron or the hole, and it actually basically hampers the recombination process, and we do not get the light we are aiming for. So we use either a light beam or electron beam to monitor the presence of these trap states. And finally, to protect the nanocrystals even better, we coat their surface with another material which protects our material of interest from environmental impact. And we use them for several applications like solar cells or light emitting devices. Now, when we are talking about nanomaterials, the first question that comes in our mind is they're so small. How do we even see it? So our eye, as we know, have certain limitation of seeing things. And then if we don't see it, the first thing we use is a light microscope. But for optical microscope, it also has a certain limitation in its resolution, which depends on the wavelength of the light that we are using. And as you can see from this equation, usually the resolution that we get with an optical microscope is around 200 nanometer. So to see even smaller particles, what we need is that we use electron microscope. So instead of light as a source, we use electron beam as a source to image the particles. And there are mostly two different kind of microscopes that we use: electron microscopes, transmission electron microscope, and scanning electron microscope. And the basic thing, how they perform is almost the same, is that both of them use electron beam to image the sample. And then this beam is passed through several electrostatic and electromagnetic lenses to actually control the shape and trajectory of the beam. But there are certain differences also. In transmission electron microscope, the sample is actually placed on the path of the electron beam. So electron beam is going through the sample, which actually helps in getting a much better resolution. And if you see this image, that this small dots that you see is actually atoms of this material. So you can see individual atoms, and with the latest electron microscope, transmission electron microscope, you can go to a resolution as low as picometer. Whereas in scanning electron microscope, it basically scans the surface of the electron, and the scattered electrons are detected by a detector, and that forms an image. So it does not have a resolution as good as transmission electron microscope, and it is mostly used to know the surface morphology. So now I would like to show you a video to just give you an idea of how much this scanning electron microscope or transmission electron microscope can zoom into one particular thing. So this is a very simple example that I found from YouTube, which is actually showing you a scanning electron microscope view of mosquito eye, and which is really interesting to see. As you can see, we are just zooming in on a mosquito in under scanning electron microscope, and you can see the range of the magnification it can go to. And you can see how it becomes more and more clear and obvious. And then they will show you once they zoom out, so you can have an idea of the scale it is zooming in. So as we can see, this microscopic world is indeed very fascinating. So I will stop this and I will show you this video, which was taken in situ inside TA machine that is actually using liquid cell TM. So basically the sample is in liquid form, and what you can see is that two silver nanocrystal here, they are coming together and they will coalesce with each other. And the size with the scale bar, you can see the size is 10 nanometer, the scale bar. So you can estimate the size of these particles, and even those reactions that are occurring in nanoscale with such small nanoparticles under TM, you can directly see them. And now you can see that they have really come close and they are coalescing together. And not only you can see that they are coalescing together physically, if you actually zoom in, you can see how their lattice planes are transforming once they are coalescing together. So I can actually increase the playback speed a bit so it will be more faster. And now you can see they have kind of diffused inside each other, and you can see that how the lattice planes are changing. So these lines that you are seeing on the screen, they are basically lattice planes of each individual atoms, and they are connecting with each other, and it is showing the rearrangement that is taking place inside the nanocrystals. And as you can see now, is it as if two particles, they are just together forming one bigger particle. And you can observe such fascinating phenomena in nanoscale directly under transmission electron microscopes. So these are some of my works. So what we do also is when we are preparing a material, as I told you already that I do shape control. How I do it is, in this particular case, I just varied the temperature from low temperature to high temperature, and I got different shapes of zinc selenide, which started with small, very small elliptical particles. Then it transformed into rod and with higher temperature, I got hemisphere shaped nanocrystals, which then further grew into size. And not only we can monitor like one particular material, I can also monitor the formation of heterostructures with TM. Now you can see that this image actually shows two different contrasts. So one part is darker and one part is lighter. So as I have said already, TM actually, it collects transmitted electrons that are transmitted through your sample. So if you have two material with different atomic numbers, the one with the higher atomic numbers, it will actually let less electron pass through it. So you can think of it as a something thick, and if light cannot pass through it properly. So electron beam also cannot pass through that sample with higher atomic number properly, and it casts a darker shadow. So you can see the gold actually, it creates a darker contrast, and zinc selenide is of lighter contrast. So we can clearly distinguish two different materials. And also, if you can see the lines in the structure, from these lines, as I said, they are basically the lattice plane. So if you calculate the distance in between these lines from the FFT as it's shown in the images, you can actually, from this distance, you can determine which material it is and which crystal phase it is. So it's very informative. And not only that, when two materials are connecting, how their lattice planes actually connect with each other, you can also determine that from the junction. So here we can actually determine that 4 lattice planes of gold at a particular direction is connecting with 3 similar lattice planes of zinc selenide in one particular direction. And that's how we get very precise information about any structural particle or any structural transformation during the heterostructure formation. So now these are also TM images, and you can take a guess of which, like what these TM images are from. So these are basically TM images collected and isolate from the first US case of COVID-19. And you can see that these blue particles, they are basically the virus. Here you can also see these are the virus particles, and this is actually the image from a single virus. And you can see that it has like a spherical body, and there are this small spike-like things on top of it. And the usual size of this virus, as I have read, is around 90 to 140 nanometer. So from TM, you can actually see those virus very carefully. So these are another set of images where you can see that the left side is a color transmission electron microscopy image, again, where you can see the body of the virus and there are kind of spikes on top of it. And this is basically an SEM image, which shows these virus particles. They are coming out of a cell membrane that was cultured in the lab. So this was basically an SEM image. So now if we go to talk about the disease, we all know that it started from Wuhan, China in 2019. And this is a typical schematic illustration of the virus, which we have seen from the structure that we have seen in TM before. So this is basically like a ball-like structure where on the top of that, you have 3 different kinds of protein, the E protein, M protein, and this S protein, which has this spike-like structure. Now, the question that came into my mind was, why is this it's called coronavirus? So I just Googled it and I found that corona basically means a part of the body that resembles that is like a crown. So this S proteins on top of the virus, they are basically look like a crown on the viral body. So that's why it is named coronavirus. And now coronavirus is not something new. It was identified in mid-1960s, and there are 4 common types, which are alpha and beta coronavirus. And then there are less common types, which is SARS-CoV, and SARS-CoV-2, which we are seeing right now. And the interesting fact about coronavirus is many of them are basically, they infect animals, but they can evolve and they can transfer from animals to human, and they can affect humans and become a new human coronavirus. The last 3 type of COVID, they are actually recent examples of this kind, where it was actually infecting an animal before, but then it got transferred to a human body. It evolved and now it's making humans sick. So as we all know, there are symptoms of coronavirus, which is very common to flu or cold. And so the very common symptoms for coronavirus typically is cough, sore throat, fever, muscle pain, chills, shortness of breath, and recently people have reported a loss of taste or smell also from coronavirus. And we also have to take care of the fact that this disease may take some time for incubation. So even if you have the virus, you may show the symptom almost 14 days later as well. As we also, all of us know that the amount of people who are being affected by coronavirus is increasing day by day. And here you can see from a simple graph that it started somewhere around January, and from June, how much the rate of infection has increased. And if you see a distribution of cases, I actually did this graph a couple of days ago. So it has even increased more. And you can see that United States is kind of providing the maximum number of infected people, whereas in India, they were almost 5% contributor in the total number of cases. And if we see for a particular country how the number of cases are increasing, you will see that initially, usually there is a high rise and eventually it reaches a plateau. And we say that reaching a plateau is good because then it can come down. And as you can see that France had kind of reached a plateau. But there are many other countries like US, which seems it's almost there, but it's still going on a little bit. And this is a graph that shows the infection in past 1 week. And you can see here the infection from India. It's actually increasing quite a lot in a steady way. So now the question is, what makes COVID-19 spread so fast? Why it is so deadly? So whenever a virus infects a human host, they have to get inside your body. And then they use actually the cell's machinery to produce copies of themselves, which will eventually go out and spread into new cells. So a study has found that the spike protein on the surface of COVID-19, the virus that I already mentioned about, it actually binds to an enzyme that is found on our outer cell membrane known as S2. So you can think of it as this COVID-19 has as a key, and S2 is a lock. So it can easily click with the lock. So it will just bind with a receptor and it will go inside the body. And in this video, you can see the same thing, that how this virus is getting inside our body. So this is what a schematic that shows you how actually the virus is affecting or getting inside our body. So now to give you a more physical idea of how the virus actually spread from one person to another. So there are basically 3 accepted modes of transmission. The first one and the most important is of course respiratory droplet transmission. So once somebody coughs or sneezes, so the droplets that come out of your mouth or nose, they can travel to a certain distance. And of course, after that, because of gravity, it falls down. So this distance is like 1 to 2 meters. And if you're there and if somebody is coughing and sneezing on you, so you will get those droplets on you, and then it may go through your nose or mouth, and it can affect you. So there comes the importance of social distancing. If you maintain a certain distance from people, maybe these droplets, when somebody is coughing or sneezing, it won't reach you. And next is airborne transmission. So you are not there when somebody is coughing or sneezing, but there are research studies that have showed that these viruses can actually retain in air. If they are forming much smaller droplet, they will retain in air for much longer time, like 2 to 3 hours, and you can get them later on. And then the third one is contact transmission. This also happens when the viral particles that are emitted from somebody, it just falls on a surface. And depending on what surface it can stay on that surface for quite some time. Like you can see for plastic or stainless steel surface, it can stay for 2 to 3 days. For cardboard, it can stay for 24 hours. For copper, for in 4 hours. And these kind of aerosol particles, as I mentioned, they can still till 3 hours. And of course, how long they stay on this surface will depend on temperature, humidity, ventilation, and amount of the virus deposited. But the key point is that the virus can stay on the surface. So if you're coming later and touching those surface, you can get the virus on your hand. And eventually, if you touch your mouth or nose, they will actually go inside your body. So currently, also, I would like to mention that there is no evidence supporting this transmission through food or water. So now, what is the importance of social distancing or the lockdown period? So there is a way to determine how this contagious diseases spread. And there is a number called R, which is called effective reproduction number, which signifies the average number of people that one infected person will pass the virus to. So for this R, it is maximum for measles. Diseases like measles, which one person can affect 12 to 18 people. For 2009 COVID, the number is 1.4 to 2.5. So if we think that it's approximately like around 3, so one infected person will transfer the, like if there are 100 infected persons, say, so they will transfer the disease to 300 people, 300 to 900, 900 to 2700, and the number will go on like that. So in 3 states of cycles of transmission, you can see the number can increase from 100 to 2700. And that is the reason why we see each day such huge increase in the number of coronavirus patients. And in this right side graph, you can see that if you change the R value a bit, how much it changes the rate of transmission of this disease. So what we try is that we try to keep the number R less than 1. So if you're not going out, if you're all social distancing, then the chances that somebody else is getting the virus from you it's actually reducing with time. So if R is 0.5, then now 100 people will transmit this disease to 50 people and 50 to 25, and eventually the infection will fade out. So that is the importance of a lockdown period. And how we can stop the spread of the germs. So these are a few instructions that are provided in the disease control website of United States. Of course, you have to maintain a 6 feet distance at least. As I have already said, that will help you not getting the respiratory droplets coming out from another person. If you are coughing or sneezing, cover your mouth with a tissue, and of course, throw the tissue in trash, and you wash your hands properly. Then whenever in public, you wear a mask covering your nose and mouth. And all these are very important to stop the spread of respiratory droplets. And when we talk about masks, we have a very important question, like what kind of mask should we use? There are several kinds. Of course, N95 is the best. It can give you protection from virus, bacteria, and pretty much everything. But N95, the production is not high, and it's mostly should be reserved for frontline healthcare workers. So the option that we have is surgical mask in some cases, which is somewhat worse than N95, but it still kind of gives a protection against virus and bacteria. But when we are talking about cloth mask, it is actually not giving protection against virus. So now the question comes is that if it is not giving me a protection from virus, why should I use a mask? The point is that it's more about protecting others. So where this kind of cloth mask is not giving you inward protection from a particular bacteria, but when you're coughing or sneezing, it's actually preventing your respiratory droplets to go out. So that way you are actually preventing the spread of the virus to others. So now we all know how to wear a mask, but I would just like to mention that before wearing a mask, we should wash our hands properly. We should always use the earlobes. We have a tendency to touch the top part of the mask. We should not use that. We should always use earlobes. And if you are using disposable masks, dispose them on a daily basis. If you're using other kinds of reusable masks, clean them properly. And after you're done with your mask, please discard them in a dedicated dustbin so that they don't end up in water bodies or cause pollution, which is I have seen plenty of images these days where these masks are ending up in sea or any water bodies, just like plastic bags, and creating huge amount of pollution. And I personally know that there are several frustrations when we wear face masks. There are like headaches, and we cannot breathe properly. People like me who wear glass, our glasses get foggy every now and then. So I understand all of them. There are certain solutions, like if you cannot breathe, find an isolated place where you can remove your mask for some time and then breathe properly. Drink more water, which can help you to reduce the headaches. You can use some anti-fogging agents on your glass, or you can also use the nose clip of your mask properly. So if you properly tight it on your nose, then maybe whatever you're exhaling, it's not coming out to making like to make your glasses foggy. And always remember that what you are doing, it's a kindness to others. You are doing it for others, if not for yourself, because sometimes you may not show symptoms. That is one thing with the coronavirus, that you may have the virus, your immunity system may be strong, you're not actually showing some symptoms, but you are carrying the virus with you. So you always should be careful. The next thing is, of course, you should not touch your mouth, eyes, nose, and mouth. You should clean and disinfect and frequently touch surfaces as much as you can. So like tables, doorknobs, switch, our computer, so there are several things that we are touching very frequently, and the virus can come from somebody's respiratory droplet and can stay on those surfaces. So we have to take care to use some disinfectant to clean those surfaces properly. One thing we should take care of, which we often don't, is that our phone, it can be contaminated very easily. So you should use some kind of disinfectant wipes, or you should really take care to protect the electronic goods from such contamination. And another thing I would like to mention, that these disinfectants, they are often nasty chemicals. So you have to be very careful reading the instructions on how to use them. There are many, I do not want to use the word stupid, but the US president some days ago, he said in a press conference that he has an idea that maybe if somebody ingests some kind of disinfectant, maybe it will go inside their body and it will prevent or it will kill the virus. And people actually followed that and they got sick. So you should under no condition eat, drink, breathe, or inject these products in your body or apply directly on your skin, as they can cause serious harm. So even don't use them on your pets. They are not for animal use. So be very careful while using those disinfectants. Next is that, of course, stay at home if you're sick. And the final and most important thing is that wash your hands often with soap and water for at least 20 seconds. So now there is another question that we often ask: is there some special kind of soap that will be more useful? So you can, of course, use hand sanitizer that has a certain alcohol content, but there is no study that shows certain antibacterial soap that are available in the market that show higher that gives us higher protection against viruses. So if you just wash your hand frequently with normal soap and water, that should be fine. And even after all these, if you catch the virus or if you show symptoms, so you should follow all those things as I have said before, but then you should stay at home. Don't go out with the symptoms if you have them. And even if you're at home and there are other people in your home, try to stay in your own room, try to use your own bathroom. And if you need medical assistance, of course, give them a call. Like, don't be afraid that they will put you in quarantine and you may undergo some problem. Always remember that if you have the disease, there is always chance that you are making other people sick, and we should not do that. So the next thing that I want to talk to you about is that now we know that the lockdown period is over in India mostly. So you can go out if you want, but the question you should always ask yourself is that, do you really need to go out? So don't go out at all if you have any kind of symptom. Always wear a mask and try to maintain 6 feet distance. Don't touch your face or mask at public places. And if you need to go to a store, bank, or other necessary services, try to choose off-peak hours because there will be less crowd. Go alone. Don't take any company if you don't need to. Don't touch what you don't need, especially when we go to shop for anything, like pretty much even vegetables. We have a tendency to touch many things and then pick the one we need. So try to minimize that. Try only touch what you need. The other question is that if we should wear gloves. So gloves can be contaminated the same way your hands can. So the best practice is washing and sanitizing your hands before you go outside and after you come back. So the next question is, finally, we have talked a lot about disease. Is there a treatment for COVID-19? So if before we go to a treatment, we need to know that how do you know, like how does a doctor know that you are affected with coronavirus? So there are two types of tests. One is one can actually test that if you are currently affected from the virus, and one it can actually test if you had the virus before. And this video will actually talk about that, how they test the presence of virus. So I hope you have an idea of how the coronavirus test is done. So next is what treatments are actually available for COVID-19. So for USA, the Food and Drug Administration, they do not have any approved medicines yet. However, FDA has granted emergency use authorization for some medicines that can be used for certain patients who require hospitalization with COVID-19. And one question that I have often faced personally, that what are the scientists doing actually? Why is it taking so long to find a cure? Aren't they working? So I would just like to point out that before a medicine or any vaccine comes to us, it has to go through several phases of human trial as it is given here. So the first stage is trying it on non-human objects. Then it is tried on a very small group of healthy people to understand that if the dose of the drug that they are giving, it's safe for human. Then it is given to a very small number of patients with the specific disease to check that if the drug is actually working. And the next stage is just a bigger thing of this one, where a larger number of actually infected people are given that particular medicine to check if the drug is having the same effect on a larger number of people. And finally it comes to the market where you can actually buy the drug with a doctor's prescription, and it actually tests the viruses the medicines, sorry, long term effect on a human body. So all these stages, they require like months to years of time, and you have to go through all these stages to get one drug approved for coming to the market and treating human patients. So right now, all the drug authorities in different countries, they have actually accelerated this process. They are trying to make it as fast as possible. and they have different programs for that. And right now, there are several anticipated drugs. So the first one that's a lot discussed, and we knew that a few days ago, US President was asking India to give them a supply of chloroquine. He was kind of threatening us, and we ended up giving them. But later on, the clinical trial actually shows that it doesn't have any actually effect that to improve the patients of coronavirus. It may actually have some adverse effects. So FDA actually stopped authorizing for emergency use of chloroquine phosphate or hydroxychloroquine, as we know. The next drug that is hopefully it can be used is remdesivir, which is an antiviral drug. And it has been observed that from phase 3 clinical trial, that the patients taking remdesivir are experiencing faster recovery as compared to patients not taking it. So this is one a very hopeful progress in that. But another question that I would, of course, like to mention here is that if antibiotics are effective preventing COVID-19, and as of now, there is no such proof that any particular antibiotic is being effective in treating COVID-19. So there was a news just a few days ago on 16th June, and came out that there are a group of researchers at University of Oxford. They're actually carrying out the biggest, one of the biggest clinical trials for finding coronavirus medicine that is ever going on in the world. And they're trying several medicines, as you see the list here. And among them, there is dexamethasone, which is just a steroid. And they treated several people with it. And they found that for people who were actually needing ventilator because they were so ill, dexamethasone can reduce the death rate by 35%, means that the doctors can prevent one death by treating 8 ventilated patients. For those who need oxygen but were not ventilated, for them also the death rate was reduced 20%, but there was no benefit in patients who didn't require any oxygen. Now, compared to remdesivir, you have to actually take it intravenous with intravenous injection, and also it's not very easily available. But dexamethasone, it's a well-known drug, it's a well-known steroid that's also cheap, and it's easily available. And shortly after these results were released, the UK government actually announced that it is immediately authorizing the use of dexamethasone for patients hospitalized with COVID-19 that require ventilator or oxygen. So when I read the news, my question was how actually this drug works. And I found out that this drug actually works by dampening down the body's immune system. And my reaction was that it is weird. Our immune system is the thing, it actually helps us fight against the virus. Once any virus or bacteria enters your body, then the immune system, it gets alert, and it actually releases a small molecule known as cytokine. So this cytokine actually triggers the cells to give a response to this virus. So stronger the response of your immune system is, the higher is it, the chances that you are not being affected by the virus or the bacteria. And that's why the young and people have lesser chances of being affected by COVID-19, because they have apparently a stronger immune system. So what happens is that sometimes in a number of cases, even when the virus is not there anymore, so say the virus is killed, the immune system still does not detect it. It keeps on releasing those molecules known as cytokines. And these cytokines, when they don't have any virus to attack, they actually attack healthy cells. And they will damage multiple organs like lungs and liver that can eventually lead to death. So it's your own immune system rather than the virus that can actually kill you. And dexamethasone, it just comes this effect. Now the question is if there is any side effect of this drug. Of course, there are. There are side effects like anxiety, difficulty sleeping, weight gain, fluid retention. But the doctor said that the amount that they're using for treatment of COVID patients, it is not supposed to bring such side effects. And another news that a very optimistic one, which just came out yesterday in Indian media, is that there is a pharmaceutical company, which is Mumbai-based, known as Glenmark. They have launched a new antiviral drug known as Fabiflu, which is kind of cheap. And it's the first Indian drug that has actually got the approval from Drug Controller General of India to be used or to be marketed and manufactured. So how this thing works is that it gets into cells and it inhibits the replication of the virus. So if you have a mild to like not mild to moderate symptoms for COVID, then this virus, Feviflu, it can actually stop the replication of the virus and it can cure it. So there are other treatments that are also available, which are blood plasma transfers that you may notice. It was talking about, the person was talking about it when we are talking about coronavirus tests. So if your body is infected with the virus, has been infected, and you have finally cured from it, so your body has produced the antibodies that can fight against coronavirus. And that antibodies from your body can be blood, can be transferred to other person to cure this infection from COVID. And there are also people who are doing stem cell research. They're also in phase 2 or phase 3 clinical trial to see if some kind of stem cell treatment can be helpful for this virus treatment. Now, there are people also researching on vaccines. So unlike medicines which you take after you are affected by virus, a vaccine is designed to protect people before they are exposed to virus. It basically trains your immune system to recognize and attack the virus when it enters your body. So there is one very useful information that you can find in New York Times. They have something called coronavirus virus vaccine tracker, and it shows you exactly at this point of time what are the stages of researches about the vaccine that is going on in the entire world. And you can see that there are at least 2 vaccines that are in phase 3 trial now. So if you go in further detail, there are actually 4 different kind of vaccines people are working on. First is genetic vaccine, where that vaccine is using one or more of coronavirus's own genes to provoke an immune response in your body. And the company that is researching more on it, it's Moderna. It's an American biotech company. So they have actually developed this kind of virus before. And right now in July, they are going to start a phase 3 clinical trial with 30,000 people. The next is viral vector vaccine, where vaccines are using a virus to deliver coronavirus gene into cell and provoke immune response. And again, this is part of University of Oxford's clinical trial, which they are doing with a pharmaceutical company known as AstraZeneca. They initially tested it on monkeys, but very soon, by the middle of this year, they are going to do human trial, and they're very optimistic that maybe by September they can have something in the market. Then there is protein-based vaccines, where vaccines that are using a coronavirus protein or protein fragment to provoke an immune response. There is again Novavax, a company based in US. They are working on it. And there is the last kind of vaccine that are actually using a weakened or inactivated version of the coronavirus itself to inject into, to give it as a vaccine in the human body to provoke immune response. And there is a Chinese company who are working on it, and they're also starting their phase 3 trial soon in China and Brazil. So now, apart from health, the most problematic factor of COVID-19 is it has so widespread impact. It has huge impact of economy, and there are 1,000 models over the internet which are explaining how it does it. But I found this one which is very simple. So if you see the economic effect of COVID-19, there is first of all, some direct costs of which increase. So you have to buy all these disinfectants and cleaning products and hand sanitizers. So it will increase your cost of living. So if you get affected, you have to get treated. So everything that will cost you a lot of money. And there are some indirect costs. Because of this lockdown period and everything, there is a loss of productivity. People are not going to work. There is a less amount of supply. So there is a problem. There is less manufactured production. There is disruption in supply chain. People cannot go to schools and colleges. We are talking about online classes, but there are many places in India where people do not have computers or even access to internet or even electricity. So forget about online classes. And it also affects mental health a lot. Nobody likes being forced to stay at home. So it eventually can cause depression as well. And then there are a lot of industries which are suffering a lot more than other industries like travel and tourism. There are retails, restaurants, entertainment industries. So combining everything, so think of all the people who work in those industries. They are all losing their job and they are becoming frustrated. And not only that, there are very small businesses. Say, let's talk about the food vendors we have usually on streets. They are not working. Nobody, even if they're working, nobody is probably going to buy food from them ever. nobody going to use street food. So there are several industries that may never come back with the same efficiency as they were working before. So it causes a lot of frustration. And typically for us, for people who are working outside India, we have a certain visa validity. So if we cannot go to the lab, if we cannot do our research, then our funding may be discontinued. And if your funding gets discontinued, we lose our job, and we cannot maintain our visa status. And eventually we have to go back. And even if you are looking for a new job, so many people have lost their jobs during COVID-19. So basically, every country is trying to give jobs to their own people. They will not look forward hiring people from other country for doing their job where their own people are jobless. So there will be a huge shortage or shrink in job market. So this is like a never foreseen effect. And I have read somewhere that people are saying that this amount of global effect is only seen after World War. World War had this kind of impact, and after that, this is COVID-19. And we do not know how far it will go. We still have no idea. So saying that, I mean, it seems too much frustrating, but I would like to say that there is still some silver lining to it, because people are staying at home, and there are less cars or transport. So we have seen much less pollution. And that's why we can see like solar system from your rooftop, and or dinosaurs have come back to the world. And from India, from Ghaziabad, you can see Burj Khalifa. From Indore, you can see Eiffel Tower. And my most favorite, from Bangalore, somebody could see Neil Armstrong footprint on moon. But jokes apart, there are scientific studies that have actually shown that there is a lot less pollutants on air after this COVID-19 lockdown. And this is a typical picture taken from a part of China where the red color actually indicates the concentration of the pollutant. It's the highest concentration. And you can see that this picture is actually, the right side one is after the lockdown, and you can see the number of pollutants is decreased a lot. And in other parts of the world, typically this is in Europe also, you can see that how much the pollution has decreased over time. So this was my 2 cents on coronavirus. I would like to finish by mentioning a very wise kid whom we made fun of. We saw Bunty in Lifebuoy ad, who used to keep saying us to wash our hands multiple times. And then we made fun of him. We said that your soap was slow. If you just use Lifebuoy, it will just be quick and you don't have to use your hands multiple times. But we were wrong. Bunty was not wrong. He was right. We should follow him. We should wash our hands multiple times for at least 20 or 30 seconds. That's the best way of protection that we can give ourselves. So take care, be safe, and be very prepared, because we still don't know what is ahead of us. And with that, I would like to thank you all for your attention. So that will be it.
Thank you, Dr. Bose, for your very nice deliberation. And as you all know, I have deliberately selected resource person from different fields. And Dr. Bose is a chemist by nature. So I am selecting few of the questions which we can answer more probably from the therapeutic approach or the bioscience is concerned. So, Dr. Bose, are you the time enough to take
Yeah,
few
sure.
questions?
Yeah, sure.
So, in your field, one interesting question is that, do you use any substrate to form heterostructures? Which type of crystal orientation you have used in the formation of heterostructures?
We do not grow heterostructures on substrate. We grow them on solution. So we do not use any crystal phase, and we just have to modulate our reaction conditions in a way so that these two different materials have some kind of chemical affinity, and they just attach with each other. So we do not use a substrate to form a heterostructure.
Do you think vitamin C will help to boost our immunity after COVID, against COVID?
I have read that vitamin C actually helps, and I have seen that in US, doctors are actually prescribing vitamin C tablets to boost your immunity. So I cannot give you any scientific like proof right now. I don't have it with me, but I think it will.
So is there any possibility for nanoparticles of screening corona out before the inhaling contaminated air?
So if you're talking about screening, it has to be some kind of a filter, right? It cannot be nanoparticles because they are so small themselves. So what you can do is that some kind of research that people are doing, they're trying to develop some particles that will actually bind to the spike protein of corona before it can bind to S2, which is found on our body. So if somebody finds nanoparticle or like synthesizes one particle that specifically targets the S protein and it binds to the protein before it can bind to S2, that may be helpful, but still nobody has done it successfully.
So plenty and abundance of comments during your nice presentation. You can also show that in the chat box. Okay, one is the TEM image related question. Spike of the virus is visible by TEM image. How much
Yes.
dense is the virus so that it gives a vivid picture of presence?
How much? Sorry, can you please repeat the question? How much?
How much dense is the virus so that it gives a vivid picture of presence?
No, I mean, once you're doing a transmission electron microscopy, you have the sample on the grid basically. So you have to scroll around the entire area to see where you can actually spot some virus. So it's kind of an optimized. You basically cannot control how much virus is there in the body, right? So they just took a sample and they placed it on a substrate, that the grid that we use for TM image, and they basically scroll through the entire grid to see where they can spot some viruses. Of course, it was dense to the extent that they can see, but it's very difficult to quantify the density of the virus. There will be places where you don't see any virus at all. There will be places where you see more number of virus. They may not be evenly distributed.
Okay, slightly off stream, but is that correct that the COVID-19 cases will be at the top in India in winter?
As I said that when people are saying that winter, there are two things. First of all, the temperature will reduce. So probably there are some studies they are saying that temperature may help in shortening the time the virus stay on the surface. So in winter, this is actually going the other way. So for summer, the spread of the virus can be a little bit less because of the temperature. For winter, it will be more. And the way it is increasing, I honestly, for like from a layman's point of view, again, I don't think it was going to reach peak very soon. So if I talk about, say, US number of cases, so they had lockdown from sometimes, not lockdown, but stay at home order from sometimes in the middle of March probably, and they lifted it sometimes in May. And they thought that it has reached a plateau and it's not increasing anymore. But even yesterday, there are 12 states in US that have recorded highest number of coronavirus affected cases. So I can say for Dallas County, when the lockdown was there, we were having number of cases per day, which was around 200. And yesterday, Dallas County has reported 418 new infection, which is highest ever. So you can see that this thing is actually increasing. And some of the people in US, they're even saying that it may be already the second wave. So if we think about India, the way it has opened up now, and the way our population density is, there is a very high probability that this will increase in a much faster way, and probably the peak is yet to come. It will take maybe another 2-3 months definitely to reach the peak or the plateau, and even after that we don't know if it will decrease. For US, I don't think it is decreasing really well.
No, plateau is not reached in case of your US or in India? In India, of course, the
That in
plateau is not
India,
reached.
definitely not reached. In US, they said the plateau has been reached. So if the plateau has been reached, so you expect the number of infections to be almost constant, little bit. But as I said, as of yesterday, there are at least 12 states in US that have reported highest one number of one-day infection. And for Dallas County, the place where I live, it has constantly seen increase in the number of particles and number of virus infected people and record number of hospitalizations. So they are saying that it has not decreased at all.
So as you lives in US, you have to defend this question. What do you think the reason behind the most medically advanced countries like USA has more death rates than in India, where medical system is not so advanced as US?
Well, I would say that it was also my idea. I would first like to say that when you talk about US, then you all think about the way it is shown in sci-fi movies, like it has all the superpowers and everything. But eventually, when the actual problem comes, then you have seen that we have such a wonderful leader here guiding the whole country who pretty much does not care about the life of people. He cares about, he is whimsical. He does not have any scientific background. He does not listen to his scientific advisors. So that way, if you run a country, it's definitely not going to fare well, even if you have proper uh, like proper medical facility. And the problem with medical facility in US also, it's very expensive. It's very, very expensive. So there are many people who may be affected and they cannot afford the medical facility. And for defending India, I would say that I think we are brought up in a way that we have a stronger immunity than these people who always, stay kind of in a more protected and cleaner environment. I believe our inherent immunity is stronger, and the policy, the way US is maintaining itself, it's way much worse. So the way it is seen as one of the powerful countries, yeah, maybe in terms of I don't know, military powers, nukes, maybe, but when it comes to common sense or common things like protecting people, they are not. At least with Trump being the president, they're not.
So is the case of our country also, but as we are public, it shouldn't be discussed. A medical perspective, this question, but as this was asked by our departmental teachers, could you please try to answer? If a patient who is on steroids for lupus treatment, which is an autoimmune disorder, and also an hydroxychloroquine as a side treatment is more susceptible to the disease or not?
It's a very difficult question to answer for me. So basically, hydroxychloroquine can only be used for some symptoms. Like if you have pneumonia, if you have developed pneumonia because of COVID-19, then there are certain symptoms like that. Pneumonia is one of them that I have read that you can use hydroxychloroquine, but hydroxychloroquine can have certain side effects if you have high blood pressure or something like that. So for the particular disease, this person is talking about this autoimmune disorder. I do not know about this disease. I do not know what kind of effect it may have on human body. So I will not be able to answer the question for certain. And I don't want to give out any wrong information.
So maybe the most interesting question, have you ever get the TM image of COVID-19? You personally?
No, I do not work on this particular field, which I have clarified before. So I am not. These are taken from CDC website, which I have mentioned in the slides. So there is a Center of Disease Control website, Public Center of Disease Control, something like CDC website in US. So they have taken these images.
So we hope we all enjoyed the session. I thank Dr. Bose for accepting our invitation. And I will finally conclude with comments from participant. Excellent presentation, Ria. How nicely you correlate your field of research to the issue of COVID-19. Very good. Congratulations. So this is
Thank
our
you so
final
much.
remarks. So uh, this is it for this session, and connect with us at the afternoon session at 5 p.m. Before you leave, I have a general comment that you have to fill the feedback form after 7 p.m. today to get participation certificate. You have to register at the lms.bgclibrary.in. So in the afternoon session, we have two speakers. One is from microbiology background and one is from physiology and therapeutic approach you will touch upon. So this is for it now. Thank you for joining with us. Thank you, Ria, once again.
Thank you.